Hello everyone, welcome to Molecura. Today we're going to be discussing the structure of the ATP and how ATP helps us to move the chemical reactions forward when we are talking about metabolism in medical biochemistry. So the first thing that we're going to talk about is going to be the definition of anabolism and catabolism. So what's happening with anabolism is going to be set of metabolic processes that build larger molecules from smaller ones, usually requires ATP. And on the other hand, catabolism is when these bigger molecules are getting broken down into smaller ones, and in these situations usually energy is released. It's important because in metabolism we're going to be talking about how energy is consumed and how energy is released through various metabolic pathways. So to understand the function of ATP better, we need to first try to understand Gibbs free energy and its implications. Gibbs free energy is a thermodynamic quantity that represents the amount of energy in a system, for example the cell, that is available to do useful work at constant temperature and constant pressure. So sometimes we refer to this as changes in Gibbs free energy, which we show it as delta G. And this is going to be in different ways. We could have the standard free energy change or delta G 0. This is going to be delta G at 25 Celsius temperature and at 1 molar concentration of substrate and product. In biochemistry, most of the time we talked about delta G0 prime, and this is going to give you the delta G0, which is the standard free energy change in biochemical condition where pH is 7. Now, how does this come into practice? Now, in general, if we want to talk about the change in the free energy, is going to be the energy of products minus the energy of the substrate. And this could have three different scenarios. The first one is going to be if the energy of your product is more than the energy of your substrate. The second situation is going to be if the energy of your product is less than the energy of your substrate. And the last scenario is where the energy of your product is equal to the energy of your substrate. So if the energy of your product is more than the energy of your substrate, it means that energy was needed to make this reaction happen. So your delta G here is going to be more than 0. So this is going to be a chemical reaction that requires energy to proceed. In the second scenario, if your product has less energy than the substrate, so the value that you're going to get here is going to be less than 0. For example, if the energy level here is 3 and the substrate had 4, your result is going to be -1. So what happens is that your delta G is going to become less than 0. And in the third scenario, of course, if the energy level of the product and the substrate are equal, your delta G is going to be 0. So in these scenarios, the reaction that is thermodynamically favorable is the one that has delta G of less than 0 or negative. This is thermodynamically favorable. So this reaction will be spontaneous. This will be a spontaneous reaction. What it means is that this reaction is going to happen, but it does not tell us how fast this reaction is going to happen. In the third scenario here, we are going to have a chemical reaction that is at equilibrium. So this reaction is going to be at equilibrium, which means that if we have a reaction that is like this, AB is getting converted into B and A, this reaction is going to be at equilibrium, which means the rate of the forward reaction is the same as the rate of the reverse reaction. So in this scenario, it's an equilibrium. If the delta G is less than 0, the reaction is spontaneous. It means that it's going to happen, but it will not tell us how fast it's going to happen. And the third scenario, if the products have more energy than the substrate, so extra energy is needed for this reaction to go forward. So this one is not favorable thermodynamically, and it's not going to go forward without an input of energy from somewhere. Now we are going to talk about these in more detail as we go and we talk about metabolism. Again, another concept to speak about quickly is going to be exergonic reactions. These are reactions which have a delta G of less than 0. What does it mean? It means that energy is getting released while the chemical reaction is happening. And in contrast, endergonic reactions are the reactions that are taking energy from its surrounding for it to proceed. So the delta G is going to be bigger than 0. So now we are going to be talking about the structure of ATP. Before we go ahead, let's try to write a chemical reaction here. So this reaction happens in our body. It's glutamic acid reacts with ammonia, and it's going to give us glutamine. So for this reaction to happen, we are going to need energy. Why? Because the delta G of this energy sits at plus 3.4 kilocalorie per mole. Now, what does this mean is that this reaction is not going to go forward unless there is some input of energy. Our delta G here is positive. What does that mean? It means that the energy that we have here in the product is more than the energy that we have in the substrate. So this energy should come from somewhere. Otherwise, this reaction is not going to go forward. There should be addition of energy to the product for this reaction to happen. In our body, this energy is going to come from ATP. Now, let's look at the structure of the ATP. So ATP is going to have 3 parts. As the name suggests, we have adenosine and we have 3 phosphates. The first structure here is going to be our nitrogenous base. This is our nitrogenous base. It's called adenine. It's a purine, as we know. And we have our ribose, which is our sugar. This is our ribose. So when adenine and ribose get attached to each other, we're going to have a structure that we call adenosine. So adenosine is basically adenine as a nitrogenous base attached to a ribose. Now, this is the first part of our ATP molecule. The second part, which is the important part, is 3 groups of phosphate attached to each other. And these phosphate groups also have names. So the first one is going to be called the alpha, beta, and gamma. And there are differences between the bonds that they make. So the bond that we have between the gamma phosphate and between the beta phosphate, and the bond that we have between the beta and the alpha is going to be high energy bonds. These bonds are going to be very high in energy, and let's try to draw it like this. And these bonds are called phosphoanhydrate bonds. These are called phosphoanhydrate bonds. Now, the energy that we get from ATP is from breakage of these bonds, because each of these bonds, when they break, they are going to yield energy. Okay, and we can write it here. The breakage of this bond is going to give you a delta G of minus 7.3 kilocalorie per mole. So the energy that we get it from ATP is going to be from breakage of this high energy phosphate bonds, and these are going to be very important when we are talking about metabolism. Now, why we wrote this reaction here? Because we're going to come back to it and we're going to say how ATP is going to make this reaction move forward. As we see here, this reaction has a positive delta G, and as we said, this is not thermodynamically favorable. But with the delta G of negative 7.3, this reaction can move forward. And how it moves forward, we're going to come to it in the next slides. One important point to have is that the structure of ATP is usually stabilized by having a cation such as magnesium attached to it. So whenever we're speaking about ATP, we are also speaking about a cation with a 2 positive charge attached to our ATP structure. So one last point to consider here is that if we remove this phosphate group, we will end up with ADP. And if we remove this 2 phosphates group, we will end up with AMP. It's also important to note that the bond between the alpha phosphate and the ribose is not considered to be high energy bond. So there are 2 bonds that are high energy, and they are between the gamma phosphate and the beta phosphate, and one between the beta phosphate and the alpha one. Now let's talk about hydrolysis of ATP. So when ATP gets broken down, and it's hydrolysis, so we will have to account for the water, it's going to be converted to ADP and phosphate. So what is happening here? This conversion is going to release energy. As we said, the phosphate bonds have high energy, and there will be a release of energy happening here. Release of energy. And this energy in our body is going to be used for anabolism, for making larger molecules from a smaller one. So here you have your anabolism. That happens as a result of using the energy that has been released from breaking ATP. And on the other hand, when we have a mixture of ADP and phosphate, we are going to have energy being made in form of ATP. So our energy is going in here. And this happens during catabolism, such as the process of breaking down larger molecules like sugar and making ATP out of it. So this is what happens during hydrolysis of ATP. Now this energy of the ATP that's getting released that we have shown here, it's going to be used for anabolism. What else this can be used for? This can be used for mechanical work. For example, when an animal needs to move its muscles, they are going to need ATP to do some mechanical work. As you can see here, moving of the muscles is going to be dependent on energy, and this energy comes in form of ATP. Another use of it is going to be transport. So what happens here is that you want to move something against the concentration gradient outside the cell, and for this you need to use energy to move something against the concentration gradient. So what's happening here, sodium is going into this transporter, but it cannot go out without the help of ATP. So what happens here, our ADP gets broken down into phosphate. This phosphate gets attached to this transporter, and the ATP is converted into ADP. Now what happens is that the addition of this phosphate to this transporter is going to change it in the structure of this protein, and this sodium are going to get out of the cell. So transport is one of the functions of ATP. Now the third function of ATP is going to be biochemical reactions. Biochemical reactions is what we are interested in this series of videos, because we're going to be talking about medical biochemistry, and namely we are now talking about metabolism. So what happens in metabolism is that we are going to need to use ATP to make reactions happen. As the first example that we gave in the previous slide, we had a reaction which had a delta G of positive. So this reaction is unfavorable. So we need to make a way to take this reaction forward. And usually this reaction can go forward with hydrolysis of ATP. And the reason is that the delta G of multiple steps of a reaction are additive. It means that we can add the delta G's together to make the reaction go forward. How this happens will be discussed in details in the next slide. Now, what do we mean when we say they are additives? Now let's go back to our chemical reaction. So the example that we're going to give here is going to be glutamic acid. Glutamic acid in a reaction in the body is going to react with ammonia, which is NH3, and it's going to give us glutamine. So what we will have at the end result is going to be glutamine. So what we have here is our glutamic acid. Here we have our NH3, and these two together are going to be called glutamine. So this reaction is important in the body for detoxifying ammonia and also to carry nitrogen through the body. So what happens here? The problem with this reaction is that its delta G is positive. So the delta G of this reaction is going to be plus 3.4 kilocalorie per mole. So what's happening here that this reaction is thermodynamically unfavorable. This reaction will not go forward without input of energy. Why? Because the energy of the product is more than the energy of the substrate. So what is happening here is that our substrate and this is our product. So the energy level of the product is more than the energy level of the substrate. So for this reaction to happen, there needs to be an addition of energy here for this reaction to go forward. And this is how ATP is going to help us. So we're going to try to draw the reaction with addition of ATP. So basically our ATP is going to get hydrolyzed. So this is our glutamic acid. It's going to be reacting with ATP. So it will react with ATP. And this reaction with ATP is going to release some energy, and what we are going to have here is going to be our glutamic acid, and it's going to have a phosphate group on it. We'll have our glutamic acid and it's going to have a phosphate group on it, and of course our ATP is now hydrolyzed to ADP. So after this we are going to have the addition of our ammonia to this mix. Now ammonia gets added to this mix, and what we're going to have the glutamine that we wanted, which is our glutamic acid plus the ammonia, and we're going to have ADP plus a phosphate group. So now we have taken this reaction and we have broken it down into two pieces. The first piece is when the glutamic acid reacts with ATP, there will be an intermediate which is glutamic acid plus phosphate plus ADP. The ammonia gets added and we have our glutamine, and then we have our ADP which got hydrolyzed from ATP. Now this reaction, if we want to write it as a whole, it's going to be like this. It's going to be our glutamic acid. It's going to react with ammonia. We are going to have ATP. And our end result is going to be our glutamine, which is our glutamic acid plus the ammonia, plus ADP plus phosphate, which got hydrolyzed from ATP. Now if we look at this reaction, as we have already talked about, so the reaction between the glutamic acid and ammonia that's getting converted to glutamine, we said has a delta G of 3.4. But as we talked about earlier, the hydrolysis of ATP, is going to give us a delta G of minus 7.3. And we talked about in the last slide that the delta G's are additive. What does that mean? It means that here we have the delta G of the first reaction being plus 3.4. This is thermodynamically unfavorable. And the second one is going to be minus 7.3. So this is the delta G of glutamine, and this is the delta G of ATP. So at the end, we're going to have, these are additive as we talked about, and the net delta G of the reaction is going to be minus 3.9. So we have taken a reaction which was thermodynamically unfavorable and we have made it favorable using ATP. Now to review this one more time, we are going to have glutamic acid and ammonia. When they want to form glutamine, we have a delta G of 3.4. Here we have the energy of the products are more than the substrate, which means energy is needed. Here we have more energy and here we have lower energy. So energy is needed for this reaction to happen. But once we are adding ATP to the equation, we are going to have the same reaction that we talked about just now, but we are having an hydrolysis of ATP happening as well. This hydrolysis of ATP is going to give you a negative 7.3 kilocalorie per mole of free energy. And since in the previous slide we said that the delta G's are additive, it means that the net delta G that we have here is going to be minus 3.9, making this reaction thermodynamically favorable. Thank you for watching. If you enjoyed this content, make sure to subscribe to our channel so you could explore more videos on Molecura.